Good morning. Thank you for coming back. We are right on schedule. My name is Tatiana Sharpy. I'm one of the co-organizers and a professor here at the Salk Institute in Computational Neuroscience. And it's my distinct pleasure today to introduce our next speaker, Christine Krug, who comes to us from the University of Magdeburg in Germany. Christine has done foundational work, so we are back into the neuroscience domain, and Christine has done foundational work from different parts of the visual system, integrating motion signals and static images. And today we will hear about the cortical codes that generates our three-dimensional experience. Welcome.
Thank you very much for the introduction and the invitation to be here. It's a great honor to be part of this quite amazing symposium. So what I'm going to want to talk about is how we generate our rich visual three-dimensional experience, particularly the dynamic one. So if you look at this video here, you can appreciate how much is going on there. You might, as an observer, think, are you at the right distance away? And if you take it from the perspective of the cheetahs running in there, they need to integrate 3D and motion information very precisely and accurately and fast in order to make the interactions work. It would probably also be harder if it was real prey they were after. And we want to look today at the cortical codes that underlie this type of visual experience, and indeed how we can generate it with artificial input. As Francis Crick aptly put it, a person's mental activities are entirely due to the behavior of the nerve cells, glia cells, and the atoms, ions, and molecules that make them up and influence them. So everything you've seen before is to do with how your brain is responding to it and analyzing it. So, but the question is at what level and how is this happening? In my talk today, I will first take a quick look at the background to the way we do these types of studies in vision and why the primate visual cortex has been such a pioneering driver to understand cognitive function. I will look at direct cortical interventions and how we can already alter complex percepts with artificial input in a very precise and predictable way. And then I will go out from these focal points of intervention and ask, how can we put maybe multiple of them together as fundamental building blocks to generate a rich visual experience. And at the end, I want to look at the question, which I think is important in order to understand the codes for perception, the way this is generated in the brain, how we can create complex dynamic visual percepts with artificial input de novo without a visual input, which has so far eluded us. So you're all familiar with this bistable Necker cube. I presume it's always the same visual input. When sometimes you see this face at the front of the cube and sometimes this one here. And this is like one of the tools we have used and others extensively in order to probe what signals in the brain contribute to perception, because it's always the same physical stimulus, but the way your visual system responds to it gives you a different perceptual experience. We can disambiguate such stimuli, and this we could here use 3D vision. Here it's just shading, and now you see one version with this face at the front here, and one with this face of the cube at the front. And this is quite important to have these kinds of stimuli in order not just to control perception, but also to control the neurons we study underneath. And the idea would be that when we look for perceptual signals in the brain, the neurons that would signal this unambiguous percept with this face in the front would also fire more when this phase is seen at the front in the bistable stimulus. And only if you get the two together, you get to the relevant representations in the brain. What has been quite important in that study is the seminal work on perceptual decision-making by Newsom, Movshan and colleagues, with very, very tightly controlled simple visual stimuli, looking at how we perceive direction of motion. So here you have a random dot stimulus where the dots, like a cloud of bees, goes everywhere. You could still try to make a choice, does it move right to the left? And here's a coherently moving stimulus moving to the left. And we can control in experiments the degree of correlation from completely random to fully correlated. And then look at how the different levels in the visual system represent the sensory information, and how this relates to when the stimulus is completely ambiguous and the percept is defined by signals in the brain. And that these kinds of stimuli allow us to probe behavior neural recording simultaneously in awake trained macaque, and all the data I'm showing you today is from rhesus macaque monkeys. That's a monkey in my lab in early training, and he is touching at the bottom, a motion stimulus comes up, and then he goes on the touch screen to signal the choice. Each time he licks, he gets a reward for getting it correct. He's actually so fast, one can't really follow it. But this has been quite important in understanding this, to be able to compare neurophysiology and behavior in real time. So with this, Newson, Movshan and colleagues have linked particularly area V5MT in the dorsal visual stream of the macaque. So here again, a macaque brain. Doris was earlier down here in temporal cortex. We're going now in the dorsal stream with V5MT lying in the superior temporal sulcus. And they linked there in a battery of tests, single neurons, to the perception of motion direction with these very simple stimuli. And they came up with a quantitative framework to do so, where they showed that the neurons there signal the stimulus, so the direction of motion. They are sensitive to the task. They discriminate the stimuli as well as the subject does. And neurons predict, the neuron signals predict the trial-by-trial percept. So single neurons on a given trial can, with a certain probability, tell you what the animal is reporting at the end, what is seen. And quite crucially at the end, all this can then, they took it together and stimulated these neurons, pushed the activity over the edge one way or the other, and could alter the perception of the direction of motion in a constructive and predictable way. This kind of way of doing the analysis has been summarized in Sense in a Single Neuron by Parker and Newsom. So what's been quite critical to understand the information processing in the brain to cognitive function has been that we can really reduce the variability and control the sensory input to the system very, very tightly, and then could probe the perceptual consequences through behavioral report. And that has allowed a detailed understanding of the distributed representations of elements of the visual world, and even to, as I just pointed out, to predict and modify visual perception. And just to, as my last slide of this historical part, what are these? I wanted to talk about what are these types of neurons and codes we're looking at. And it is quite interesting that in the Barlow 1972 paper, Sense in a Single Neuron, Horace Barlow made a prediction of how this should be. He predicted these, asked for these experiments and predicted what kind of code we're looking at. And this is reverberating through a lot of our work on many groups. And I think most of these things have held true in these tests. So he postulated that if we want to look and understand nervous function, we need to look at the interaction at the cellular level, not more macroscopic or microscopic, because this is how you get signals across the brain in these intercellular interactions. He postulated that the sensory system should achieve as complete a representation of the sensory stimulus possible with a minimum number of active neurons or sparse code, that these trigger features that are matched with patterns of the visual stimulation are shaped by experiences, developed processes. There's plenty of evidence for that, how they're shaped through this. And that when we look at perception, we should look at neurons representing something of the complexity of the event symbolized by a word. So it could be left or it could be right, for instance. And he firmly put his mark on a rate code of high impulse frequency correspond to high certainty, and as the trigger feature is present. The key ideas I want to take out of this is we need biological plausibility in the code, and I don't need to say this to anybody here, but it's spikes and action potentials that would convey a signal from visual cortex into motor structures to get a response. And the other thing is that is probably the most controversial these days when we can record from so many neurons at the same time. He predicted there would be sparse coding and cardinal neurons, not at the level of the grandmother cell, but at some intermediate level. And that, I think, for visual processing, we most likely find in mid-level visual areas. So this was random motion. Can we go and account for more complex percept where we combine multiple cues, an object perhaps, and alter that with our understanding of the circuitry in a constructive way? And we're going back into V5MT in the STS for looking at 3D and motion integration. It gets input from earlier visual areas with the right information. We know for the tasks we're looking at, there are no perceptually relevant signals in there, but the sensory information is there. It's tightly connected to parietal cortex for the sensory motor transformation, and it also gets input from reward representations, for instance. And I will show you that artificial signals in V5MT can shape visual perceptions. I will touch briefly on, this is also the representation where you find reward expectation signals, and they can change perceptions reacting through there. And in other works, we have shown that social influence affect visual perceptions through these representations. So this is my stimulus, and now it takes audience participation. You have to wake up. So, right, okay, who sees the front surface of this rotating cylinder going to the right? Hands up. Who sees it going to the left? And who has not seen it switch yet? Okay, you need a bit more time. I can reassure us that there was a paper by Jack Pettigrew that if you can't see these stimuli switch, you have sticky hemispheres and more likely to have bipolar disorder. I can assure you we disapproved this in a published paper. I can give you the reference, so this is not true. Anyway, this is a bistable stimulus of random motion. It's actually two transparent planes of random motion going in opposite directions. There's no 3D depths in there. It's all in your brain that you see the rotating cylinder and it flips.
I see it as flat. They're just going back and forth with the sign.
I think I have to have a word with you afterwards, but... No, it is very difficult with the different size screens. I must say the Solk is one of the biggest screens I've dealt with, but it's also one of the most beautiful. So if it's smaller, it's better. Okay, so as I said, there is no depth information in there, but it should flip at stochastic intervals. It's a bistable stimulus. It's actually one with a very, very long persistence rate. So on average, it should be 10, 12 seconds for most of you. We can disambiguate this figure by putting the left and the rightward moving dots at different depths. So for instance, if we put the leftward moving dots in the front and the rightwards one in the back, you get a clockwise rotating cylinder is viewed from above, or basically the front surface to the left. And conversely, if you go the rightward moving dots in front and the leftward in the back, you get a counterclockwise rotating cylinder. And you can smoothly titrate this information to get a nice psychometric function. Neurons in the area V5MT have a neural representation for binocular depth and direction of motion. So we again here, this is a slightly unfolded macaque brain. Here's the STS. It's a highly myelinated area V5MT, and neurons are tuned exquisitely to direction of motion, and about 80 to 90%, and then 2/3 of them also to 3D depths, and this one here to near. It's mostly near or far. And with that, neurons in V5MT have a receptive field that is located in space in the contralateral hemisphere, prefer direction of motion at one depth. And so here, this is a view from above, the neuron would respond to clockwise rotation, but not counterclockwise rotation, even though by themselves the same depth and motion information is in the receptive field, just in a different conjunction. So the task for the monkey is as such, the monkey would have to fixate on a fixation point. We have mapped the receptive field. The cylinder comes on in the receptive field, and at the end of the trial, the monkey has to make an eye movement to indicate its perceptual choice to the left. You've seen it rotating the front surface to the left and to the right, And what you get is very nice psychometric responses. These monkeys can do the task very, very well. This is a single neuron recording, pretty representative. They're as good as the monkeys on a lot of the time. And when we ask for the bistable cylinder stimulus, which is here at 0 depth, 0 binocular disparity, and ask whether on trials the monkey chose the preferred direction of the neuron clockwise, it would fire more than on counterclockwise. So whether the variance in the firing here predicted perceptual choice, you get really nice, clear correlations. So this is the data from a single cell from that here. This is a trial number. These are only the 0 disparity trials, the bistable one, and we plotted spikes per the 2-second trial, and you can see the preferred choice here in green and the null choice in blue, and you can already see in the marginal histogram that there's a clear difference trial by trial. And we can quantify this difference, this link with perception. For those of you familiar, it's termed choice probability, with which an ideal observer could predict the monkey's perceptual choice based on the firing of this neuron alone. And in this case, this neuron is at about where the average of the population for MT is, at 0.67. If you would take 4 neurons with independent information, you could account for the perceptual choice of the monkey. So it's a very close link. So, but this is a correlation. So are these neurons really contributing to perception? So if they do, if we push them and give a few more spikes in these neurons, we should be able to change the percept. And this is work by a very talented former doctoral and postdoctoral student in my lab, Nela Sismil. So we went in and did a focal electrical microstimulation at sites in V5MT. And of course we knew that when you electrically stimulate there, work by Dan Salzman, you can increase motion perception for the right, and work by Greg De Angelis, you can increase perception of near versus far. But both of these by themselves would not change in a coherent way our cylinder percept. So if you increase motion direction to the right, it would be at the front and the back. You get no rotation gain. And if you just do near, it would be potentially also for both directions. So the question was, can we interact with that structure in V5MT in a way and add spikes that would just be read out as a conjunction between, for example, right and near, and get more counterclockwise rotation. So the experimental task was the monkey fixates. Again, we this time plot the multi-unit receptive field where we put the stimulus, visual stimulus in. At the end of the trial, the monkey makes an eye movement, reports that the perceived direction of rotation, and he gets rewarded for correct choices. Only rewarded for correct choices with regard to the visual stimulus, not with the microstimulation. So we give the animal every incentive to break or find something wrong with the electrical stimulation. On half the trial, randomly interleaved, we inserted a very small current microstimulation through a single electrode in one focal point in V5MT. And this is the result from one side here. You have the psychometric function with the cylinders. Again, the disparities down here. That's your bistable cylinder. Counterclockwise rotation in one direction, clockwise in the other. And these are the trials where there was no electrical microstimulation. And now we insert We place the electrode in the center of a column, match the cylinder stimulus to the receptive field properties, and stimulate with 20 microampere biphasic pulse. And what we see in this site that's preferring counterclockwise rotation is that for every given visual stimulus, we find more choices counterclockwise rotation, so in the preference of the stimulated site. And not just the bistable stimulus, the ambiguous one, but here stimuli the animal gets 100% correct when there's no electrical microstimulation. And here the animal actually loses rewards when it does so. We can quantify the effect of microstimulation by the shift in this in the function here in terms of how much visual information we would have had to put in order to get the same effect. And it doesn't just work for stimuli in the front or at one direction. So here's one example. So this is a side that prefers right and in front, and you get more rotation direction in the counterclockwise rotation. This is the shift between the non-stim and the stimulated trials. This is a different stimulation side which preferred right and far, and electrically stimulating this, you get a shift of the perceptual report in the opposite rotation direction. So we can specifically interact with the representation in V5MT and make the rotation direction one way or another, depending on where we stimulate. And it's predictable from the preference at the site of the multi-unit activity. Yes, when it's and it's highly constructive, so you don't see any deterioration in the percept, as flattening of the slopes. We also compared the size of the shift, and we did this as a microstimulation. The shift I've just shown you over the sensory threshold to be comparable. For our stimuli here in red, the disparity motion stimulus from a different study from Greg De Angelis, where he only did microstim for motion and depth. The sites were similarly tuned and selective. And you can see either if you take all data or if you take just the side with a significant effect, though while he finds significant shifts with microstimulation, our more precisely matched stimuli to both motion and depth to the representation of V5MT gives you the bigger effect. So the stronger is the effect. This is also the site, I want to touch on this briefly, where we get reward signaling. And it shows, for instance, in how expected reward at the end of the trial, so whether the monkey knows he gets a small or a big reward, how that affects the microstimulation effect in V5MT. So the idea was if reward is all to do with parietal and higher up, the shift in the sensory signal would be the same. So you wouldn't get an effect of stimulation over non-stimulation overall. But what we see is when expected reward is small, the microstimulation effect is larger on the same site. That's a single experiment again, than it is on the when the expected reward is large, which suggests an interaction between the two, and that here the sensory gain to the expectation of large reward is turned up. And that's been confirmed by drift diffusion modeling of our results, that our results are most likely explained by an increase in sensitivity in the sensory neuronal pool. So while it shows that there's the site here, it also helps us potentially down the line in microstimulation experiments, because here we made the reward act against the stimulation, but we could also help entrain the system because these signals are also there. So I've shown you some evidence that focal microstimulation in extrastriate visual area V5MT can alter complex percepts made up of two visual cues. The alteration is predictable and constructive. It's likely to add spikes in the neuronal pool. Precise localization of the visual stimulus and the task relative to the stimulated neurons is essential. So small focal currents are sufficient to activate defined elements of the visual model. So we can change the rotation direction of that visual stimulus. But what we've not been able so far is in these higher visual areas, and this is also in human experiments, to really create complex stimuli from scratch. We still need the visual stimulus in all these experiments in order to get the structure of the whole visual stimulus itself. So what else would we need in order to take this node we have? How many of these would we need, or what other signals in order to create that complex visual stimulus from scratch? So what are the minimum building blocks for our rich visual experience? And I just want to stress this is not about replicating the whole brain network in the state it sees that percept, but asking what is the minimum number of nodes we need signal to generate this. So one thing, if you look at V5MT in terms of the anatomy, and that's work by Bashir Ahmed, with whom I've worked over 2 decades in my lab in Oxford. So if you look here, this is a schematic of a flat mount through layer 2, 3 in V5MT. This is an injection site of CTB. And all these dots here are cells across the whole of extent of V5MT that have a synapse at this site and project there. And you can see this clustered projection all over. When we do quantitative analysis, what we find is that there's a regularly 2 millimeter repeat, which is quite interesting if you think about the columnar architecture of V5MT. It has columns for the same direction, as shown by Tom Albright, and then clusters of 3D depths over those by De Angelis and Newsom. The direction columns have a repeat of 1 millimeter. The depth column is about 0.3 to 0.7 millimeters across. So we don't connect every direction cycle, but potentially a network of same disparity and same motion with each other. And one idea would be to go into these. And these clusters here, if you go in a parasagittal section, here's the injection site, you can see they form columns along at regular intervals. So they go through the cortical layers. And that got us also the idea of, obviously, we have this, perceptual signals for motion and depth in V5MT. And we can stimulate and get alter the percept, but is this really a common processing pathway, or can we learn something about the dynamic interactions in these circuits? And so this is the random motion stimulus and the choice and the rotating cylinder. And I just put the number for the link with perception at the bottom. That's the work from Britton and Newsom with 0.56, just about significant over 0.5, and a much stronger link with perception of the cylinder. And we went to explore to look within the local columns at the neuronal interactions. And we did this first by looking at spike count correlations between the local single unit and the multi-unit at the same site for the rotating cylinder, the bistable one, and the random motion stimulus. And so you can see in this example that the correlation is much stronger. They share a lot more noise. They're closer linked to each other for when the cylinder stimulus is on than for the random motion. And this holds across the sites for 2 animals, particularly when the sites are strongly tuned. And when we look at this shared noise, the local interneural correlations, where the single and the multi-unit are more strongly linked, the single unit has a closer link with perception, again, as choice probability. And we can look at the time scale of these interactions within a cortical column and can see that of these interactions by integrating across the correllogram over time, and can show that the difference in the interneural correlation between the cylinder and the motion arises on a longer time scale. There's nothing to do with synchrony and is 10 milliseconds, 20. It arises from about 20, 30 milliseconds, so a difference to about 100. And these interactions, these rising interactions in the first half of the trial, as here on the y-axis, when you plot them against the choice probability, the link with perception, how it arises, this signal over time of the trial, a rising phase, a stronger rise here is correlated with a stronger link with perception later in the trial in the signal. So 3D motion columns are interconnected in a specific pattern across V5MT, form a network. CD cylinder and random motion have different signal dynamics, and the neural interactions within a column on a long time scale between 3 to 200 milliseconds are linked and predict a perceptual signal. So, can it tell us how we have to integrate these sites across nodes potentially in order to arrive at the percept, and how to recreate it? So, of course, I've only talked about V5MT here. So when we talk about the whole decision process, we have a whole arc coming from sensory information represented in V1, where others have shown there are no perceptually relevant signals to our task. V5MT, where we have the perceptual evidence, decision variable and LIP, and then transformation when an eye movement is made into in these to the saccade response. And of course, there are other areas involved there too. We and others have looked at some of these representations. This is work from Andrew Parker in relationship with in collaboration with John Reynolds at the Salk. We looked at the moving waves and data from our lab from arrays, and in this case in V1 and V4, that shows traveling waves and how they might connect different layers in the network. And I think these interactions in the sensory areas will be important for us to know at what state we need to take the cortex into when we apply the artificial stimulation to be effective. We've also looked at higher up at lateral interparietal area, which is getting input from V5MT as well as other visual areas and connect to the frontal eye fields. What is quite interesting, they have both sensory representations and motor response field, these neurons, and you get also quite tight sensory responses to disparity and to motion. So, where do we stimulate and at what spatial pattern? I focused on V5MT because I think it is a critical point. We have already these points with which we can change the perception. If you inject the tracer here, I've shown you, this is another view of this pattern in the flat mount, you get this interconnected network, and it's most likely we have to go across the column and stimulate columns with the same preference to get a coherent percept in the absence of a visual stimulus. But we would also need to look, obviously, at other connected points, though I don't think one would need this resting state network centered on V5MT. One would need to have all these areas, because there's a critical representation here in V5MT. With electrical microstimulation, we just put a current in. We don't know which cell types we stimulate. So one way forward would be potentially to look at that. We just, for example, with optogenetics, stimulate excitatory neurons, NV5MT, and see whether we can get a better output. The other question is that of temporal correlation, and I don't think it will be that we have to stimulate them at the same, just at the same time, but the interactions and the timescales will be important within here and potentially with higher areas. And again, optogenetic should provide the tool that we can vary the temporal signal much better in a much more targeted way, based on what we have seen with the interactions of neurons that are relevant to perception. So what is the minimum artificial signal to be planted into cortex to generate a complex percept de novo? I think it's critical to identify the local spatiotemporal dynamics of neuronal activity that define the specific percept. We will need multi-site electrical and optogenetic stimulation targeted to the relevant sensitive sites. We need to understand the temporal patterns to kickstart and fully engage the specific circuit, and potentially use contextual factors, neuromodulators, to boost the signal, change the cortical, the brain state, utilize system spasticity for readout, but the price for the stereo motion is potentially, the reward is quite high, because the structure for motion objects, they're not just there. I mean, it would help us understand how these objects are formed, but it's also about navigation in space, how different direction of motions are integrated at different depths. So if we could learn to stimulate this, not only would we gain some insight in the circuits and codes that underlie our rich 3D motion perception and these objects, but also help us potentially generate neural prosthetics that allow us to navigate through space potentially not swing through the trees, so like the orangutan here, but that is a critical signal to move through any kind of dynamic space. I've mentioned most of the people that were involved. Just want to say that all this work has been in collaboration with Andrew Parker in Oxford and now in Magdeburg as a senior professor. And funders are importantly the Wellcome Trust and die Deutsche Forschungsgemeinschaft. And this is my group waving from a balcony saying happy birthday. So thank you.
Thank you, Christine. So we have time for maybe 1 or 2 questions. Maybe more questions.
Hi, that was really lovely and impressive work. On the initial stimulation, it looked like you saw a very clear sort of effect in the bias of the animal's response, and I would have predicted actually that what you might affect in the areas where you're stimulating was going to be something closer to a change in sensitivity. And I'm wondering if you sort of have an idea about underlying computations that might be manipulated that would lead to a change in bias and not necessarily, or maybe there is a change in sensitivity that just wasn't clear in your slides. And if you could maybe expand on that a little bit.
I mean, there is no change in slope between the non-stimulated and the stimulated trials, but there are some experiments where the effect at the beginning is so huge that it really drifts apart, and then the animal, it centers. So it's not that there is no bias, if you've seen on the non-stimulated trials, and there's only bias in the microstim, they shift. So the animal tries very, very hard to maximize the rewards, but cannot dissociate the two signals. So what we think we're doing is adding really activity into this pool, adding spikes into these neurons. And it really only works when you very closely match your stimulus to the multi-unit receptor field. So there's another group in the US who's tried this experiment before us and couldn't make it work. And they didn't generate the stimulus in the experiment. They had preset movies, and then it doesn't work. We looked at early and late phases of the stimulation, and we don't see a change there. It can wear out, but it's not, there are experiments where it just simply doesn't, and I can't tell you why. So, but basically it shifts apart, and then the monkeys adjust these to come back. So they try hard to break this, but they can't.
Thank you.
Wonderful talk, Christine. So I have 2 questions. One is that your bistable stimulus, I think it's very analogous to this rivalry stimulus that I showed. So have you looked at decoding over time? You know, is there any evidence for this biphasic representation of the conscious percept and physical stimulus in your data?
We have done some decoding, and we do not see this going up and down. We see a rising phase early in the trial, and then it stays at the level of V5MT stable over the course of the trial. We have some more data. I mean, this is what I've shown is some of the single neuron data, single recordings. We have some with high density electrodes where we're looking at, which would probably be easier to see, but we don't see it in the single neurons. No, it's a very stable signal once it's established, which fits with the stability of our percept. I mean, there's one fundamental difference to the rivalry in our stimulus. So there is no information that tells the animal it rotates one way or the other. There is not a suppressed stimulus, if you want. So in the percept, basically all the dots are subsumed of it. It doesn't have to suppress part of it.
Right. Okay, second question is, like, you talked about how you can, affect a very specific percept or create a very specific percept. So, like, you can have very tiny cylinders, right? And they can be moving independently. I know these MT receptive fields, my cursory understanding is that the receptive fields are quite large. Mm-hmm. And so how do they encode, small moving things that are moving in opposite directions? And when you stimulate like an MT column, what happens to the percept of like multiple small things?
Multiple small things. So obviously the neuron is sensitive in the receptive field to small patches of direction of motion and depth. we haven't explicitly probed smaller one, which was an interesting question, but what we have looked at is actually the neurons are sensitive to the relative depths between the front and the back plane. So this is, I don't think it's about small objects, this is about bigger planes and the difference between them at different depths. But I guess what you would want to ask is I put something here and something there at different depths, how that would integrate, and I can't answer that at this point.